occurs. So we said that sulfated, these polysaccharides, which are sulfate containing or sulfated polysaccharides, they are different, even with the closely related species. But here's just example. So 3 different species, 3 different sulfate containing polysaccharides, but they all are very different chemically, right? So we are going to give an example of sulfate, what we call fucose sulfate. And based on this, we are going to describe the mechanism of acrosomal reaction. Okay. So fucose sulfate is present in none of this. Actually, it's present in Strongyloides purpuratus species. So one of the species, just one of the species, just think of the sea chain. Okay. So the interesting thing is that we are saying that fucose sulfate is different from different species. That means this is one of the barriers for only the specific sperm of a specific species can enter it. Now these are secreted by the egg jelly. So egg jelly is going to secrete it. And how this process occurs? So if you remember, we said right here is your sperm membrane. And beneath the sperm membrane, right here is your acrosome vesicle. Okay. So the fucose sulfate that is released from the egg jelly that start this reaction of what you call acrosome reaction. So the receptor for the fucose sulfate. Now fucose sulfate is in the egg jelly. So egg jelly is secreting fucose sulfate. Okay. And fucose sulfate should come and bind to the sperm, right? So there should be some sort of receptors. So receptors for fucose sulfate are present right at the membrane, right here, above the acrosome vesicle. Okay? So right here, so this is then, right here are the receptors for fucose sulfate. Okay. Now, what is going to happen? So I'm just drawing it a little bit bigger. So this is the area where these receptors are. Okay. So now what is going to happen? So this is the receptor right here. So let's say this is the whole big membrane. Above this is the receptor. Right here, the fucose sulfate come and bind. So that's the first step. So we are having fucose sulfate receptor binding. Fucose sulfate receptor binding. When fucose sulfate binds to receptor, what that does, there are certain calcium channels. So these are calcium channels. These calcium channels, they open. So this is binding, and that binding leads to a calcium channel opening. Step 2. First step is receptor binding to fucose sulfate, and when that bind, that leads to opening of the calcium channel. And when that happens, from here, the calcium ions start moving in. And when the calcium ions start moving in, right here, along with this are present right here is present sodium proton antipolar. What are antipolar?
Like when one comes in, one goes out.
One comes in, one goes out. Okay. So sodium potassium antipolar are sodium proton antipolar are present. This is happening in all the cells. Whatever cell you talk about, human, monkey, donkey, sea urchin, everywhere, sodium concentration is high outside. And in case of sea urchin, specifically because outside is ocean, right? So sodium concentration is high outside. When this is going to open, what is going to happen that from here, sodium ions are going to move in and proton ions are going to go out. So proton is going to go out, making it more basic inside, okay? And sodium is getting in. Now, because this thing is happening, this whole activation of getting sodium in, getting potassium, calcium ion in, that is going to activate a membrane-bound enzyme. Right here is a membrane-bound enzyme. So this enzyme is phospholipase C, also you can say PLC. So this phospholipase C enzyme is activated, okay? So this is ultimately, both of these things, what they do, they activate phospholipase C, okay? When this is activated, what it does, it converts phosphatidyl inositol biphosphate. So this is a lipid, phospholipid, that is actually attached to the membrane, okay? This is also membrane-bound enzyme, so this is also present in the membrane. And what it does, this is also, you can call it PIP2. And this enzyme, when it activates, it converts this phosphatidyl inositol biphosphate is converted to inositol triphosphate. This is called IP3. Okay. Now, when this thing happened, this IP3 is considered as a major regulator of calcium ion concentration. Okay. If you have more IP3 in the cell, that means you are generating a huge amount of calcium flux inside the cell. Okay. So what that does, IP3, it causes more calcium. Okay, and where this thing is happening, everything is happening here, right here. Everything is happening right here, right? Right above the acrosome vesicle. And remember from your cell biology classes, we said more calcium causes exocytosis. Okay, so what is going to happen? More calcium, that is going to trigger exocytosis. So what is going to happen? Because of this vesicle which was here, acrosome vesicle, this vesicle is going to come here and fuse to the membrane of the sperm. So then what is going to happen? Now you have a condition like this. So this acrosome vesicle content is released. right? So everything is released outside. Now what is going to happen? So we are saying that so what happened? The first part of acrosome reaction has taken place. So what is going to happen? This fusion of acrosome, so that is triggered exocytosis, that means the fusion of acrosome vesicle with sperm membrane has taken place. Okay? Now what is going to happen? This is there, this is going to basically start digesting the egg jelly, right? So it's digesting the egg jelly, but the second process was, it should not be like this, we should have a acrosome process also coming out, right? So right here what was, right here you had G-actin. You had the globular actin there, okay? Now more calcium, what it does, it activates what we call RHO-B, R-H-O, RHO-B. So RHO-B, what is RHO-B? RHO-B is a GDP binding protein, and we know from our cell biology that means this is GTPase. That means it will start activating when it is attached to GTP. Okay? So what RHO-B does, once it is active, starts polymerization of actin. So now what is going to happen? So this actin is start going to polymerize, and when this actin is start going to polymerize, it's going to form like this, it's going to push it up, and what that is going to do is that it's going to form a right here, the acrosome process is going to form. Okay. So what we are saying? We are saying that the acrosome reaction is mediated by sugar that is sulfate contained. And we said different oligosaccharides, sulfate containing oligosaccharides, they are different in different species. Okay? And we have given a special example of one of the species of sea urchin. In that species, fucose sulfate is that polysaccharide that is sulfate containing polysaccharide. And we are saying that fucose sulfate bind to receptors, and those receptors are located directly above the acrosome vesicle in the membrane of the sperm. Okay? When fucose sulfate bind to this receptor, that immediately initiate the opening of calcium channels. These calcium channels open that allow calcium to enter in. That start a cascade of process, and the first process there happen, the sodium proton antiporter open. Sodium gets in, protons go out. Change in pH, entry of calcium, entry of sodium, activate the membrane-bound enzyme called phospholipase C. When phospholipase C is activated, it basically converts phosphatidyl inositol biphosphate PIP2 into inositol triphosphate. Inositol triphosphate is a major calcium regulator in the cells. Once its concentration up, that is going to increase the concentration of calcium. And what is happening everywhere? Right above the acrosome vesicle. And we know that higher calcium concentration is going to initiate the process of exocytosis. So what is going to happen? This acrosome vesicle is going to move and basically fuse with the sperm membrane. Once it fuses, acrosome vesicle release all its content outside, and then they start digesting the matrix. And then high calcium, what it does, it activates RHO-B. RHO-B is a GDP binding protein, that's meant it's a GTPase. Once it is active, it starts polymerization of actin, and polymerization of actin is going to form a actin microfilament, and that actin microfilament is going to make a acrosome process. So this is how the acrosomation takes place. That is making sense? Okay, talk to each other for a second and we'll go further.
Remembering the acrosomal vesicle, and then there's the receptors for the FS on the outside. So FS binds to its receptor, which causes the calcium channels to open, which then causes the sodium hydrogen or antiporter to open, which initiates the phospholipase C to activate. When it's active, it converts phosphatidylindosylphosphate biphosphate into inositol triphosphate. IP3 causes more calcium. And then that triggers phagocytosis, diffusion of the basement membrane, and the acrosomal vesicle contents are around. And then because of the more calcium, RH or Rho-D is also activated, which starts the polymerization of ether G-actin, and then that I think the more calcium goes, they're together. But then also, how does it make more calcium? It just always channels open.
And this one? This is a big break.
So when you say IP3 causes more calcium, is it just it helps keep the channel open?
Yes, IP3 actually binds to the calcium channels. In our cells, or most of the cells, the calcium is stored in the endoplasmic reticulum. And there are receptors on the endoplasmic reticulum, what are called IP3 receptor. IP3 bind them and open, so calcium comes out. Any doubt here? You guys are okay here? Okay, now. So now we have talked about two things so far, which are species specific. Number one was reset, right? Sperm activating protein. Then we said acrosome causing carbohydrates, right? So far two things. Then we are at the third thing, that is recognition of eggs. Okay, good. And what is that? White line in the middle, right? In our case, Jonah pellucida, right? So that is the extracellular outer coat, or I should also say extracellular coat, because that's outside. Right? So, so what is now happening? If you think about it, you had now a sperm, and this sperm this is the acrosome process. And right here was the jelly, right? Inside right here is this is your, right here is your white line inner, right? And inside right here should be this should be your egg membrane. If this is egg membrane. Making sense so far? So what has done? It has basically digested the matrix, Now the acrosome process need to make contact with white line envelope so that it can digest it and go in, right? So this need to reach here to the egg membrane. So we said that white line envelope is digested by the proteasome which are present on the acrosome, right? But it can digest only of its own species. Why? Because it can only contact to the white line envelope of its own species. It means there should be something on the acrosome membrane that can help the recognition of the white line envelope so that it can bind to the white line envelope of only of its own species, nowhere else. Okay? So what they have found that on the acrosome process, there is a special protein. That protein is called binding. Binding proteins are present. And these binding proteins are kind of like coding this whole thing. So binding protein are present. So if I show you here, so right here is the immunostaining for the binding. So this binding is present on the acrosome process. So if you take that binding from different species, for Aspergillus, as Francinus, whatever it is. So if you have binding protein, now you take an egg, de-gel the egg, basically remove the jelly, right? And you put the binding protein. We said this binding protein help the egg to bind to the white line envelope, right? If you take binding protein from the same species, then you can see this binding is binding to all the eggs. But if you take binding protein from different species, green is different species, this doesn't bind to egg, right? So it means binding is very specific for species specific. So same binding can bind to the same species of the egg, and binding is present right here on the acrosome process. Making sense so far? Now, so binding is usually a large protein. These are 30,500 Dalton. That's the molecular weight. Those are the big, large proteins there. And so, if we are saying the binding is there, right, then there should be some receptor also which can be recognized by the binding, right? So what they have found, if you have, this is for example the egg, okay? Now I'm not showing you any matrix, I'm showing this is the white line envelope I'm showing you, right? Inside is the egg membrane. So this is white line envelope. This is so if you take an egg and you basically throw sperms there, millions of sperm. So what happened that these sperm, they do not bind everywhere. You will find many sperm binding like this, many sperm binding like this, many sperm. So they only bind to a specific position. They don't bind everywhere. Okay? So that means that there are special places on which these acrosome process can bind, and acrosome process have binding which allow it to bind, right? And the receptor for this is called receptor protein for binding is EBR1. Okay? Now EBR1, thousands or hundreds of EBR1 molecules, they are clustered like this. So here you will have thousands of EBR1 clustered like this, and they cluster together to form the receptor of binding. Okay? So hundreds or thousands of, usually it is hundreds. So hundreds of EBR1 molecule will cluster together. So for example here, if this is the place, you will say, okay, here, hundreds of EBR are clustered together, hundreds of EBR clustered together, they are clustered together, they are clustered together at the spaces. So these are the EBR1 clusters right here. To which the binding which is present in the acrosome process bind. Okay. Now, this is even in the closest species, this is important, even in the closest species of the sea urchin, where every protein matches, even in those species, the sequence of ABR1 and binding will be different. So that means that these proteins, binding and ABR, these proteins are actually fastest evolving protein. Because everything is same. But these two proteins, they are fastest evolving proteins. So that means these proteins are responsible for act as reproductive barriers. So they are acting as reproductive barriers. And when we are saying they are acting as reproductive barriers, that means they are playing a role in sexual selection. That is making sense? So that means what is happening there, we are having, at least we have talked about three barriers there. We said attraction, species-specific attraction. Then we said the polysaccharide which are causing acrosome reaction. And even if that happens by chance, this is a third barrier which cannot be broken. That is making sense so far? So this was about how the white line envelope is recognized. Now white line envelope is recognized. What are here? We said there are proteasomes there. So these proteasomes, what are they going to do? They are going to help this acrosome process now to move to the egg membrane. That is making sense so far? So far the picture is clear, right? Okay. So then let's go to the next thing. And that is fusion of egg and sperm cell membrane. Now think about it. So this was your sperm. Okay. Right here was the nucleus. And think about the egg. So right here is the white line envelope. And it has now digested the white line envelope. And now it has reached the egg membrane. And this is nucleus. Right? So far is okay? This we just talked about. Now, this egg membrane is in contact with the sperm membrane. Here, contact doesn't mean that membrane are going to fuse, right? If you remember from your previous class of cell biology, we said there are snare protein which allow to come in very close proximity so that lipid bilayers can basically mix with each other. So it means there should be something here which can allow it to mix with the lipid bilayer of the egg membrane, right? So it means there should be something. So we are saying the fusion of sperm and egg membrane is mediated by what we call fusogenic proteins. Remember, membrane is what? Membrane is lipid and proteins, right? So it means lipid is there, but the protein should also be able to get in so that those membranes can fuse with each other, right? Here in this case, in this particular species we are talking, as Perchlorantherus, in this species, binding acts as fusogenic protein. How it occurs? N-terminus of binding has many hydrophobic amino acids. So it has many, and those are long stretches, has many. Essentially, they are long stretches. So think about that it's a hydrophobic protein. Hydrophobic proteins, can they easily interact with the membrane protein and the phospholipids? Can they do? Right? Talk to each other and give me the answer. Don't look at me. Just talk to each other.
Can you repeat the question?
So the question is, hydrophobic proteins or hydrophobic amino acids, can they easily interact with the membrane proteins? And can they easily interact with the phospholipids? If they can, why, where? If they cannot, why, how?
Even if they were on the outside, they're still rooted in there, and this is still gonna be okay.
Okay, what do you guys think?
Well, you kind of said that the membrane is part of what the membrane does, so it kind of interacts, but we're not really sure if it's going to be actually.
What do you think?
We said that we didn't think it would interact with the proteins, but we thought it could interact with the membrane.
Okay, let's think about this. So proteins can be like this, protein can be like this, and proteins can be like this.
So we don't think it'll interact with the ones that are mostly just on the outside. Maybe
So
we
it cannot interact on this part of the protein, but this part is hydrophobic, right? This is polar, but this is hydrophobic. It means those proteins can actually interact with phospholipids,
Okay.
can also interact with the hydrophobic portion of the protein in the membrane, not this side. So that means that can allow the mixing of the membrane. Making sense? Yes or no? So what that cause, because they have these long stretches of hydrophobic amino acid, that cause actually mixing of the membrane and fusion of the membrane. So it fuses like this. Okay. And if you look at the picture, this is picture is showing you like this. So you can see here, kind of this is entering to the white line envelope, right? Once it's there, this is kind of in the membrane they're showing. So it's kind of the fusion of the membrane has taken place. Okay? So the fusion of the membrane takes place. Now, remember when we were doing the structure of the egg, we said right here are also beneath, right here, throughout the egg, we said there is actin, G-actin. Remember when we were talking about the structure? And we said that also polymerize and give you microvilli like this. So right here, where actually the fusion took place, there was a microvilli right here. Right here. And that caused the fusion. Now, here is actin on the egg side, right? Right here was also actin on the sperm side. Cytoskeletal protein are there, right? So these cytoskeletal protein, these actin from egg and sperm combine to form cytoplasmic bridge. Actin can combine to each other, right? They are the same protein. Polymerization is going to take place. Now what happens? So the fusion of membrane takes place. First thing, because of the fusioning protein binding, and then you have actin combined together like this, they make a cytoplasmic bridge, right? Then more actin start coming together along with this, and widen the cytoplasmic bridge. Making sense? So this was kind of this, then it widens. So then cytoplasmic bridge widens. And when the cytoplasmic bridge, so this whole thing is not going to enter. This is just going to stop here. What is going to happen from here, once this bridge is formed, kind of your path, right? Through which nucleus, haploid nucleus, and centriole, those two things enter in the cell. Haploid nucleus and the centriole, that's all. And once that happens, so far everything is clear. If there is any doubt anywhere. Okay, now this enter, now what is going to happen? So now you have this cell, you have a centriole. That centriole came from sperm, right? Then you had a centriole that was present from the egg, but that degenerated. We said earlier also that that centriole from the egg will degenerate, right? So this is the centriole from egg, but that degenerated. So it will actually go away, it will be no more. Okay? So we have only centriole that is present from the egg. So this is a haploid nucleus coming from sperm, haploid nucleus coming from egg. Haploid, and they are going to come to end, right? Now, all the mitochondria which were present in the middle piece, they never entered in the egg. So all the mitochondria we are getting there from the mother. Okay? All the centriole we are getting there from the father. All the cytoplasm we are getting there from the mother. Everything except a nucleus and centriole is coming from the father, everything else is coming from the mother. Now what happens? This centriole is going to divide into two. Centriole divides into two. So one centriole here, one centriole here, start moving on both sides like this, and immediately the cell division start. So this nuclear membrane go away, as the cell division happens, right? Nuclear membrane go away, chromosomes are now free. For example, 2 chromosomes were here, 2 were here, right? And total 4 chromosomes become, diploid number become. That is okay, everyone is understanding. This is simple mitosis, right? And these are going to divide. This centriole come here, this chromosome will kind of like become this, like this. And ultimately you will have 2 cell form. Each cell will have 1, 2, 3, 4, 1, 2, 3, 4. Right? Because 2 chromosomes coming from male, 2 chromosomes coming from female, 4. And then mitosis takes place, chromosome number remain same. Any doubt here? Any doubt? Yes, no?
Does the genetic material like mix them together?
Say it again.
Does the genetic material like mix them together? Or
Yeah.
just-
Because as they enter the membrane of the nucleus break, how that happens we talk. But I'm just giving you this, like once it enters, mitosis start. And the zygote, which has become 2N, will produce 2N cell, right? We are all 2N because gametes were haploid. So what is happening? Haploid gametes, NN, sperm, egg, fuse. Zygote, 2N. The zygote divide, give 2 cell, 2N, right? That's what it happened. And we always get 2 and 2N. We are all diploid. Now what happens? So far we are very happily talking about fusogenic protein. We are saying they are helping the sperm to fuse. Millions of sperm are being produced. Millions of sperm will be attracted, right? A million of sperm will fuse. So think about all that thing which were helping it to fuse is now can become liability if one sperm has already fused it. What will happen? Just think. If instead of 1 sperm, 2 sperm have entered inside. Now think about instead of 1 sperm, 2 sperm have. So this is the nucleus. These are, for example, the 2 chromosomes of the egg. Okay? You have the 2 chromosomes of the egg right there. And then, right here, 1 sperm has entered. This 1 sperm has provided 2 chromosomes and 1 centriole. Then let's see, another sperm has entered. That sperm has provided 2 chromosomes and a centriole. So what is going to happen? This centriole is going to divide into 2. Okay. This centriole, which is right here, is going to divide into 2. So in a cell, so this is 1 cell, in this cell what is going to happen, you are going to have one centriole here, one centriole here, one centriole here, one centriole here. Okay? Now there are 6 chromosomes there. So you will have 4 cells will be formed. One might have two chromosome, one might have only one chromosome. Right? So from one cell, 4 cells will be produced, and they will all have different number of chromosomes. Or 2 may have same, 2 may have different. So what is going to happen? This embryo, if it starts developing, it will degenerate. It will never develop. But that's going to create a lot of problems. So nature doesn't want that, right? So that's why we do not want polyspermy. So polyspermy means more than one sperm. So then they have the mechanism to avoid that. So what are those mechanisms? We'll talk in a minute. But before that, let's read about what we have just talked about. From the recognition to fusion, just read and see if there is any confusion, then we'll go further. Any doubt? Any doubt here? Okay. So now what happens, we are saying that binding is a liability, right? We should block that not more than one sperm can enter there. So there are two mechanisms there. The first mechanism is called fast block to polyspermy. Fast block to polyspermy. And this actually happened within 1 to 3 seconds after first sperm binding. Okay? And this is achieved by, fast block is achieved by changing the membrane potential of egg membrane. Okay, what happens here? So if I'm writing here, on this side, I'm writing here the voltage. And that voltage in millivolt. Okay, here is the time. So if I take an electrode and put that electrode, what is the resting potential of our cells usually?
Negative 70.
Negative 70. So it means that egg membrane has a potential of negative 70 millivolts. So this is like this. Will always be negative 70, right? And say here that this was negative 70, and what happened is sperm enters here. This is the time when sperm enters. The sperm binding. Sperm binding. Immediately when sperm binds, within 1 to 3 seconds, what if this is here, this will go up. And this will go up, and it is going to reach plus 20 millivolt. So this will go there, and this will reach plus 20 millivolt. And what is going to happen after that, it will stay there almost for a minute and then calm down. Okay. Now, for a minute, this is just a 1 minute time. After this, it goes up 1 to 3 seconds, then stay up for a minute, 1 minute, and then go down. So what happens? From minus 72, it reaches plus 20, right? So the fusogenic protein, which are there, the sperm fusogenic proteins are positively charged. So when the egg membrane is negative, those fusogenic protein the sperm can come here, and those fusogenic protein can help to fuse because the membrane is negatively charged. But what happens when the sperm is right here, fuses with this, binding takes place, that opens, so binding of sperm and egg opens sodium channels on the egg membrane. And when that sodium channel are open, what happens? That leads to influx of sodium ion. And influx of sodium ion that raises membrane potential from minus 70 to plus 20 millivolt. Okay. And when that happens, you're going to change the membrane potential. So membrane will become positively charged. So the sperm cannot fuse. Okay, so that's the first line of defense happen. Now, if you take a egg and you artificially raise the charge, say take an egg and make it plus 20, then the sperm cannot bind. And if you artificially, you forcefully does not allow, for example, you put an egg in a sodium-free environment. For example, take a sea urchin egg is in the ocean, right? Instead of that, you put that egg into, for example, into fresh water. There is no sodium outside. So then the polyspermy cannot be blocked. More and more sperm can be blocked. But think about this I'm talking about saying this mechanism is there. But think about, for example, fishes, freshwater fishes, amphibian, they don't live in ocean, right? They live in the fresh water. In the fresh water, there is no sodium outside. Then how they protect it? So in that case, what happened, the chloride channel opens. For example, in amphibian, which are in fresh water. So what will happen if chloride channel open? What will happen? So chloride will go out of the cell, right? Chloride concentrates are high in the cell. So inside negative charge will increase or decrease if chloride goes out. It will become more positive, right? So chloride start going out and it becomes more positive, and that is called as, again, the fast block to polyspermy. Now, this fast block to polyspermy is a critical step, but it's only for a minute. A minute is good enough, but not good enough. Then there's another mechanism which is called slow block to polyspermy, right? So talk about this fast block, then we'll talk about the slow. Talk to each other about that.
It happens in 1 to 2 seconds after sperm binding, and it's a chain containing membrane membrane. So sperm different proteins are properly charged. When the sperm binds, it shoots up to plus 20 for a minute. The binding of the sperm egg opens the sodium channels of the egg membrane, which increases the influx of sodium, which raises from negative 7 to plus 20 or less. Fresh water is the chloride channels, which makes it rush out, which makes it possible.
When, does this start taking place like right when it touches like
Yes,
the
yeah,
vivilion?
yeah,
Okay.
right here. Once it touches infusion start, it does not want any other sperm to start that process.
When it touches the egg or the vitamin?
So white envelope is only gone, right? Because it is happening all in the egg. Because binding starts with egg membrane, right? White envelope is just a cross. White envelope has nothing to do with this. This is all egg membrane. Because there is no cytoplasm between white line membrane and ink, right? It's just secreted out. Any other question here? Okay, so we will keep on continuing this. I think we don't have time now. We'll talk about slow block to polyspermy and go further. And hopefully our cells will be here today, they are saying. So we are on for Friday at 4:30.